Yeah, hello and good evening out there. So I hope you hear me well, and I hope you can see my screen already. And Marcus will tell if this is not the case.
All is good.
So thanks, Marcus. Thanks, Marcus, for the kind introduction. And since you're probably all or spread all around the world, so I basically start with a picture of Hamburg. So we are very far in the north, and here on the picture you actually see what Hamburg is especially proud of in the moment, which is the Elbphilharmonie, a very new building right at the Elbe River. And this made Hamburg very well known. And just to show you one more picture, so everybody in Hamburg was really proud to see that the New York Times listed Hamburg as ranked 10 of the 52 places you should visit in 2017 with this very nice picture of the Speicherstadt. So we will talk, or I will basically talk about work we are doing with protein structures, and there's also another strong link to Hamburg. So here on this picture, you see the European XFEL, which is a free electron laser, which is currently built underneath of Hamburg. So it's in a 3.4 kilometers long tunnel, and it's a laser which produces 27,000 pulses a second with less than 100 femtosecond lengths. And so it has a brilliance which is about 1 billion times higher than a regular conventional X-ray source. And although I'm not an experimentalist, I find this really amazing. And if you talk to the scientists who work with this machine, so they hope that in the future they will be able to make a single non-crystalline molecular structure determination and even produce time-resolved structures so that you get into really measuring or recording molecular movies. This will certainly take some time, but I think this is a very good point actually to start, because we will see that with machines like this, but also with X-ray facilities around the world, we get more and more structural data. And this is a picture you probably see in many talks. It basically shows the growth rate of the PDB. So we all know that that we have an increase in growth, and I just want to mention that I assume that the increase will be even higher in the next years. So protein structures are used in drug design for I don't know how many years. It has a long tradition. And what typically happens is we start with individual protein structures and start to model, use them as the basis for rational design of new ligands. We will connect, of course, this field of individual structural modeling in this talk. However, I also want to point out what we can actually do, or what we should be able to do with these large amount of structures. So how can we actually deal with more than 100,000, probably in a few years, more than 1 million different structures? And how can we use this information, and take knowledge out of it for our individual projects? So this will play a certain role. Okay, before we go there, I basically want to show you where we are and what we basically do in Hamburg. So my academic group here in Hamburg actually focus on the method development somewhere in cheminformatics or structure-based design. So this is the whole spectrum we are doing. And furthermore, we also have a lot of interest in working on interactive and visualization-based problems. And in this triangle, you find lots of tools we developed over the last now 20 years. Here's just a list of names, and I will not go through every of them. So some of them are well known. FlexX is basically where everything started here. And the thing which is important for me is that we are not doing just academic software for fun. So I think a project for us is only successful if we find some people out there who are really using this program in their practical work. So we have different channels where we really try to distribute the software. And you see, basically, we do a lot with Biosolve IT, and we also have a suite of software tools you can directly get from the university. And we have a web service where we actually start to add more and more structure-based services too. And actually, this web service will also be part I will show you during this talk. So since we have the focus on structure-based design, we will very much here on the right side of the diagram. Nevertheless, I will show you just one very quick slide for one of the tools on the left, so to just get an impression what we do and why we do it. Okay, so yeah, as I promised, just one example, one slide of one of the cheminformatics tools, which is the Smarts Editor, which is actually a tool I'm really proud of that we developed it and that we have it. So it's a good example for a university tool because it originates from from teaching, where we basically had the problems to train the biochemists and chemists in how to create a smarts expression for searching molecules in databases. And from that, a graphical language was developed, and from the graphical language, an editor was developed so that these patterns can now be drawn. And with the drawing of the pattern, we also developed now possibilities to immediately see the matchings in different molecule sets. And the most recent thing we added to this is that we now, with a certain graph mining technology, are also able to automatically create patterns which separate molecules from each other. So this is basically which shows perfectly the breadth of the methods we are doing, from visualization ideas to interactive user interfaces to really tricky cheminformatics algorithms here, which is this graph mining approach, in the hope that we can produce something which is helpful for people who have to solve cheminformatics problems. Okay, but with this, I want to switch, and we will go to the real structure-based design field, and I will start introducing you to a software technology, which is, first of all, a desktop application again, and the technology is a database technology to deal with protein structures. So the software tool itself is named Pelican, and the side tool of this is Naomi Nova. But now I will basically talk about the Pelican. Okay, so what's the Pelican doing? It looks like it's basically eating structures, and that's the idea. So Pelican is a big database which handles all protein structures. However, in a way that you can actually search it in a slightly different way, which is probably usually known to you, by using PDB or other services. So the Pelican is an approach to allow you or to enable you to completely search in a geometrical way. So here are a few questions which might be interesting to answer. So you might look at a certain structure, and while you're looking at it, you see that there's a certain functional group, and you might ask whether the hydrogen bond geometry you see is very reasonable, whether there are other examples for this kind of hydrogen bond. You might ask, if you look at certain atoms, maybe a halogen, whether the distance you see in a structure is very typical, which give you an idea of energetically favorable interactions. Maybe you ask whether certain functional groups are in the proximity of other amino acids, whether you see this in other structures, or you basically ask for what you basically find in similar protein environments, what kind of structures you find there. So, and all these questions have in common that you basically ask a geometric question, not a question related to the sequence of the protein or textual information. This is all basically geometric. And these kind of questions are not so easy to solve and to find out quickly. Some of you might know ReallyBase, which actually was, to my knowledge, the first tool who was a technology to answer these kind of geometric questions. I knew this from the very beginning because this was a project where I was also involved. I have never done this, but I was involved into this. So, and we basically were thinking about whether we can do it more flexible, faster, and easier to use in a kind of a desktop application so that everybody can just ask these questions. And here's just to start with a way how this actually works. So although you can create queries also from scratch, the typical scenario is that you have a protein ligand complex in mind, and you start actually to define a geometric query. So the query can contain a certain number of atoms, which are these green points now, which are all selected. It looks a little bit like a pharmacophore, but it's not. So it's basically that you select atoms on the protein side and on the ligand side. Between these, you can define distances, and each of these points actually can also be annotated with lots of properties. So you can basically say whether it's part of the protein, part of the ligand, whether it comes from a certain amino acid, from whether it has a certain atom type, or whether it just is a kind of a acceptor. You can actually even define smarts expression if you have an idea that the atom should be in a certain chemical environment. So once you have done this, you can add distances between them and say that you want to have a certain geometric arrangement between them. You could also add angular information. So for ring systems, we know already a direction, which is just perpendicular to the plane. And also for hydrogen bonds, we have directional information, and you can also define angles here between these distances. Our method also contains all kind of textual information, and the good thing now is that you can combine these things. So you can basically combine these kind of queries with whatever you like. You can ask for certain resolutions for an experimental type, maybe for a certain range of EC numbers for an organism or whatever. Yeah, and then you can basically ask shoot out the query and get the results. So the technology behind it uses more or less all of our chem informatics platform we have in the background. So we start with the PDB structures, we extract ligand molecules, we have the framework to actually reliably extract the chemical structures, including protonation states, tautomeric forms, etc. We use now the technology to define binding sites around these. We now extract with some kind of text mining all the textual information from the PDB file. We have lots of descriptors we calculate for the pocket and the ligand so that these could also be integrated in the query. Then we take near every atom we find in the binding site and annotate it as a potential search point, that you can actually start your search over there with annotations on the type and where it comes from. We pre-calculate a lot of interactions and store all this in the database. What happens here in the database is something which would take quite a long time to explain. So Therese, so a PhD student who has done this work, she basically spent more than 2 years just to optimize the technology behind this database to really get a fast geometric search in here. So instead of going into too much technical detail, you can read it if you like. This was published this year. I just want to show you the power of such a technology. So let's assume you have a structure like this. This is a typical geometric challenge. So you see a so-called aromatic cage, which is very well known, for example, for factor 10A. So you have these 3 phenyl rings here, which actually form a cage, and you typically have small ligand atoms in here. So that is a good case for a geometric search. You actually can define these atoms. You could also add then distances to define the shape of the cage, and you could add angle constraints to make sure that the phenyl rings really point to the inside of the cage. And once you have done this, you can basically do the search. So we have a subselection of the PDB actually with about 60,000 structures, which these are all structures where we have ligand atoms in and which are interesting. And from these, we are actually performing the search. So the search takes now 25 seconds. And within these 25 seconds, you find about 200 matches, which come from 66 PDB files, lots of hydrolases, several factor 10A structures, but also a trypsin structure. And you can look at all of these and basically see what kinds of ligands are in the pockets, what kind of atoms are formed there. So we have mostly nitrogen here sometimes, but also a carbon, and analyze the structures. So that's basically the idea, how we basically get this information out of the PDB. Another example here, that was at the time when we developed this technology, we read a paper where the whole paper was just about what kind of interactions do chlorines at phenyl rings make to other phenyl rings. Is there a preferred perpendicular orientation of chlorine atoms? The paper describes a lot of scripting technology to get this information out of the PDB. And while we read this, we constructed this very easy query here with some distances. We go to the PDB, look at the 60,000 structures, need about 70 seconds to find all 213 cases where we see this geometric arrangement in the PDB. Yeah, so, and this can be analyzed again. So you couldn't look at all of these, but you also can create statistics. So for example, you can look at the distance. So what's the typical distance between the chlorine atom and the phenyl ring here down? Yeah, so there's indeed a characteristic here at about 3.8 angstrom. You could also look at the angles. So the angle here at the chlorine atom or the angle here at the phenyl ring, and look at the statistics there if you see a certain preference for a pattern. So I think this is a very reasonable and useful tool to get just information from the PDB and to gain knowledge of what's really in this wealth of structures, which is probably more than what an individual chemist or modeler have in mind from their personal experience. Okay, so with this, we also, with this database technology, created another tool, and I will not talk about this very long. It was basically a side product since we were interested to optimize our hydrogen bond geometries. So we created a tool where you can actually define any kind of functional group by so-called smarts patterns you've seen before. You can then map them over the whole PDB and create distributions of interacting atoms around it. So the kind of pictures you get, so this is just part of the process, which is probably not so interesting anymore, but the kind of pictures you're getting are these. So you have a certain functional group, you see the distribution of oxygen, nitrogens around it. You can have density plots, and this is also similar to tools which are already known. So CCDC has tools like this. We basically developed this because we want to have the full freedom of defining the functional groups the way we were interested in them. And furthermore, what we are interested in is to have a fully backlinked interface. So which basically means you can click on every oxygen or nitrogen or sulfur atom here in this cloud, and whenever you click on this, it will basically give you the structure behind it and will show you the geometric arrangement of the functional groups. Yeah, so you basically click on it and then you get this information. You can also project this information into the protein structure. We use this already for extending, for example, a ligand by seeing what kind of interactions we see in the proximity of a known ligand and then actually extending the molecules in a certain direction. All right, so we use this technology to actually derive new geometries for hydrogen bonds. I will not talk about this in detail, but there was a JMAT Chem paper earlier this year, which have lots of plots like these ones here, where you see the geometric preferences. So these kind of studies were already done at the time when the PDB had about 30 structures. Yeah, so where people try to find out what's the preference for certain kind of hydrogen bonds. Yeah, and with some longer time periods in between, we have now redone this study so that we have now the information from 10,000s of interactions between these functional groups in protein ligand environments, and we can look at the geometric distribution we see, for example, here for the carboxylate, for in-plane to out-of-plane angle distribution. Okay, so here's the test case or the application where we look at an extension. So you basically have a protein ligand complex over here. You project these interacting spots here around these functional groups, and then you can actually click on these, and basically you get via the backlink, you get the protein ligand complex, which is behind these certain structure. You get an idea of the functional groups which are placed in this region, and this might help you in extending molecules and getting new ideas of how to design or change a molecular functional group here to get new compounds. All right, so that was basically one part, and from here on I will switch to a series of tools which we developed with basically one goal. We want to be able to really go through the whole PDB, and in a fully automated fashion, we want to get all information necessary to use the protein ligand complexes for molecular docking or modeling studies. If you say that this is already possible, it depends a little bit. So our aim is to have it in a very high quality and definitely without any manual intervention. Because for the application you have seen just with Pelican, we cannot afford to look at all protein structures and fix hydrogen problems, for example. Yeah, we basically need this all done in a completely automated fashion. So the first tool I want to show you is EDIA, and EDIA has the aim to evaluate the protein structure and measure how well the electron density is there to support the structure model you'll see in the PDB. So here's an example. You have a certain side chain in the protein, and you might use this coordinate information here for your modeling tasks. Yeah, when you do that, you basically rely on these structural information. You basically rely on these atom positions here. And although crystallographers know very well that there's electron density behind it, and this is just an interpretation of the electron density, this is often forgotten when you basically just open the PDB file and you look at the molecules. Yeah. Our aim was to develop a measure which actually is able to tell you individual for every atom how well this is really resolved in the electron density, so that you get an idea from the modeler's perspective how reliable the atom position really is. So if we have this scale here, so this is the EDIA scale from 1.2 to 0, so everything which is blue is very good, and here it becomes more and more fuzzy. So this is the coloring you basically get out of it. And if you now look at the electron density at the 1 sigma level, you really see that that the side chain here is by far not clear whether it's placed at this side or not. And we believe that this is an extremely important information for modeling, and it is of course also very extremely information for us as a method developer, because we trust on these structures when we try to find new ways of dealing with protein structures, of docking and scoring, for example. Okay, so for EDIA, just a short idea how it works. So you probably think there are B factors out there, there's an RSCC, there are already several measures, and it would take quite some time to go through the full discussion why one or the other is better or not. You can look at this paper here, which gives you the information we looked and compared a lot between these things. I think the most important difference is that EDR is not a comparison between two maps. So most methods are based on the idea of putting a structure back to a map and then compare a map to a map. So what we do is we oversample the grid. We have a certain weighting factor, which actually within an atom, it gives you a benefit if you have electron density there, and it gives a penalty when you are in the proximity of the atom. Yeah, this would not really work because we have covalent structures, and being on the side of the atom there's another atom. So the trick with EDR is that we have in this third step here an algorithm which makes an ownership assignment. Actually, that means for every very small voxel of electron density, we decide to which atom this really belongs, and this takes place under consideration of the covalent structure of the molecules in the background. Yeah, then this data will be accumulated and gives you values for each individual atoms. Here's just a plot to give you an idea what's the difference between RSCC, for those who know what this actually is. It's a very well-known measure for the crystallographic quality of the structure, and here's the EDR. And these are the calculations of all atoms of proteins in the iridium HT data set. You see that there's a high correlation in here, which is a little bit blurred out because of you can't see the density of these dots here. But the thing I want to show you with this diagram is that we do not only get an idea of whether the atom is well resolved or not. By analyzing the environment around the atom, we also get an idea what's going wrong here. So you see the different colors of the dots, and you see here the meaning of these dots. So you basically have lots of atoms where we think that the density is a little bit low, but you also have situations like here, these orange ones, which basically show that for these atoms there's a shifted density. So it means there is enough density in the proximity, but the atom is not centered in the electron density, but shifted to the side. Yeah. We also see clashes of atoms with not enough density or with unaccounted density in the direct proximity, which is also a sign that maybe one of the atoms has to be shifted. So we can actually do an integrated error analysis in a fully automated way while we look at the EDR values. Just a few examples here, so we can now look at lots of structures. So a sidechain here with EDR values of 0.4, which is very low, and an RSCC of 0.8. The reason why this happens is that RSCC measures correlation, EDR measures the absolute availability of electron density. So you see there is density in here which supports this direction, but this is certainly not at the level which we would expect it if we have a clear signal for the structure here. Yeah, and yeah, lots of other cases we can look at here. This is again an interesting case here where we basically see that the EDR value is high, but the RSCC is low. What we see is that we have a little bit untypically shaped electron density, but since EDR just accumulates electron density in the atom vicinity, it's not that critical that the shape is somehow looking a little bit different. Yeah. So this is what happens if you look at ligands. So EDR values are, for most of the cases, very good, yeah, but you have outliers. And I basically want to show you a few of these outliers you actually can see. So to start with something good, so here is a structure of a ligand molecule where we have an EDR value of 1.09, so a very high value. This is a perfect structure. You basically see every atom. There's no doubt about the exact conformation of the small molecule. The resolution is also, of course, very high. But this is not always the case. So we have situations like this where most of the structure is very well resolved, but you have flexible chains which are simply not possible to have them just modeled by one individual structure. And you basically see this also in the EDR value. This is something you would also see in the B factor, but you actually see it also here. Here it gets already fuzzier. We always have structures here with the resolution better than 2 angstroms. Yeah, and you have ligand molecules like here, where basically parts of the structures are missing, where it already becomes tricky to really be sure about the orientations of side chains. Here are 3 cases, all with a resolution of 1.9 in the structure with an EDR of lesser than 0.2. So where we basically see that larger portions of the structures are not resolved by the electron density. And here is actually the point reached where we are extremely careful in using these kind of structures. For example, if we validate a docking method. All right, so that was EDR, which is actually the first thing to do when it comes to the analysis of structures. From there on, we have to look at hydrogens. We have Protoss developed already several years ago. I will not talk about this in detail because this is already 3 years old. Protoss actually assigns hydrogens, it flips side chains if necessary, and it also corrects tautomeric states to actually get the right hydrogen bond interaction pattern in the protein-ligand interface and in the protein at all. So Protoss is very fast, so usually we can run it easily, so it's usually in the millisecond range, so we can easily run it during we do calculations like docking or scoring. Yeah. So if you look at it, you see actually this is also from 2014, so I don't want to know how these values for the other tools would be now, but if you just look at the Protoss values, you basically see the importance of really have tautomeric and protonation states, which is the difference between the right and the left here, which basically show you that there are lots of cases which cannot be handled without a full integration of tautomerism into the process of hydrogen assignment. Okay, so the next interesting problem is that of binding site alignment. When we looked at this about now 2 or 3 years ago, the first time, we always thought that this is more or less solved. But then we started to look at structures and at alignments we get from modeling tools, and we were rather frustrated. So situations where ligands are sitting in the interface between different domains. Situations where we have homodimeric structures, where we have just 2 different orientations of the same protein and cannot distinguish them. Situations where you have large arrangements, rearrangements in the active site. And in all these cases, it's extremely difficult to get really very good, high quality alignments which are centered off the active site. So that was the reason why we developed a new approach for that problem. This is active site centered, it has a residue mapping, it has an interface handling, so it doesn't matter whether you are between 2 or 3 interfaces. It allows multiple matchings of the same structure, and it matches very flexible regions reliably, and you get the right results. Yeah, so that's basically the idea of Ascona, the alignment tool. And also, due to the lack of time, I will not explain you how the algorithm really works here. Just to show you that it really works, so if you take the STEX non-native data set and you basically take about 50% of them which have an annotation of the domains, and we can basically compare actually the predictions with the assignment we have for the active sites, you basically see that in nearly all cases we make the assignment correct. There's only one case actually this is the same. It's just 2 different structures of the same scenario where we have a deviation here from the PDB annotation, and these are cases which are like here where the domain annotation in the PDB is inconsistent. So in one case we have the active site between A and C, and in the other one we have it between A and D. Yeah, this causes the differences here in these calculations. Just to show you that we can also make a right alignment in the cases of strong structural deviation. So this is usually the case where structure-based alignment will not work anymore. Yeah, because we have a huge rearrangement of amino acids here in the active site of the protein. So this is integrated into a complete pipeline. I mentioned this already. We are interested in such a pipeline, which goes from active site screening. So we actually can search from one protein to the whole set. Then the alignment, we can superimpose them in a fully automated fashion. We optimize the hydrogen bond network, and we also have several tools available for ensemble reduction, so that with one click you can go from a single structure to a protein ligand centric ensemble automatically created from the PDB, so that you can really study protein flexibility, mutations, and also use this for docking with the integration of protein flexibility. Okay, so with this, I want to show you a little bit of the web server, and the name of the server is Proteins Plus. It's part of the German Network for Bioinformatics Infrastructure, so we get a little bit funding of making this fancy.
Yes,
so yeah, so as you probably see now, we are not in PowerPoint anymore. So you basically see the Proteins Plus in my web browser here. And I just selected one protein yesterday evening. I just looked around and thought what might be interesting. So I choose a metalloproteinase protease with an inhibitor structure, which is 2-OCR, OZR. And this is basically the way Proteins Plus starts. So it basically starts with a protein structure. And from here on, you can basically start and use all these tools I just mentioned. So the first is here on the left. You basically see the protein ligand complex. I have to give all the credits for this amazing viewer actually to the PDB people themselves. So Alexander Rose from the San Diego Supercomputing Center from RSCB, he developed this viewer here, and we basically just use them. You also know it probably from the PDB. So just to give you an idea, so there's a viewer here. In the middle part, you basically see the ligand structures, which are already ran through the Naomi pipeline. So they already have the correct assignment of the bond types, etc. The protonation took place. So here you see the neutral form of the ligands. You can basically browse and see all the ligands which are in the structure. You can actually click there to just have a look and see it here in 3D. On the right side, you basically see here the information for the PDB code, and you see link outs to the PDB, but also to the Brenda database. So if you are interested in getting enzymatic informations on that, you can go to Brenda here via the EC number. So yeah, so let's look at some tools here. So first of all, I mentioned Protoss. So all the tools were here on the right. You can do a Protoss calculation here. And once you've done this, you actually see already the hydrogens here on the ligand side. You don't see them on the protein, basically because we have the cartoon representation. I can go to the lines mode, but then it basically, of course, will get difficult to actually see all this. There's, in this example, there's not a very tricky tautomeric zone or so that's not in here. But as you can imagine, you can just use this to get an idea of the protonation. So as with all tools, the information is downloadable, because we know that this is, we call this our modeling support server. It's not a modeling server. So you basically can use this information, download it, and then continue working with other tools. That's always the idea. We spent time in the past to work on active site prediction. So I limit this basically to the just one of the tetrameric protein domains here. You can actually calculate active sites in here. You can visualize them here. You can also get all the statistical information about the pocket, the distribution, the properties, the distribution, and you get the well-known druggability score we calculate for the pocket. So actually, the dockside scorer is the most frequently used service from the Proteins Plus. So this is already very widely known, probably because there are so few services which do druggability estimation in the PDB. So what hopefully some of you remember is from probably also looking at the PDB is that you can get post-view pictures. So these post-view pictures are from us if you have seen them before. So post-view is also linked. So if you just want to get a nice picture, if you want to see the hydrogen bonds, etc., you can use post-view to actually go there. But also the tools we have seen in the talk before are linked to Proteins Plus. So here's, for example, the EDR. So EDR is the electron density quality estimation. So we can basically look in here. So you get now the different coloring. Yeah, you get the coloring of the EDR values. You basically have the color scale here. You see there's something going on in the structure. It is in fact a structure with 2.3 angstroms resolution, so it's not surprising to see this. And what you basically can do, you can see here all the values for each individual amino acid. You can also look at the values for the individual ligands, and basically, for example, you can pick the one with the best EDR value to start working on because you have the best resolution in here. If you're interested in one or two, you could also get a view to the electron density, and basically to compare this, you can actually see, for example, here that really electron density is missing at this part, which caused the red coloring in the ligand at this point. Okay, so from here you can also go to Sienna. So we can actually go to Sienna. So Sienna actually does this active site search, and we have lots of parameters to control. We found out that this, for most users, this is too complicated, and we basically hide all these parameters and just have the typical settings. For example, you can say, I want to compare ligand poses in the active site of this protein. You basically select the ligands which you're interested in, and then you can do the EDR, the Sienna calculations. So this typically takes about 10 to 20 seconds. This was now a little bit faster because we always cache results for about 2 weeks, and I have done this yesterday evening. So you basically see all the PDB files which are available for this structural element. So where you have an identical active site here in this region, you can look at these and you can overlay them. Yeah, and while you do so, you actually also see alternative ligands which are placed here in the active site. So this is, for example, I think a quite interesting scenario, because you see that there are ligands which fit to the other part here of the active site. So this is just one case. You can, of course, click at a lot of structures. Here you see, for example, an alternative fragment in a small molecule, which represents an alternative to the triple bond we have in our structure. Yeah, which nearly perfectly aligns in the active site with the ligand structure we started with. So, so this gives you lots of ideas. These tables are all interactive, so you can search, for example, for the structure with the strongest deviation in backbone, yeah, which would be this one here. Still a ligand which totally goes to the other side of the active site, interacting with the metal ion over here. So lots of things to explore, yeah. And of course, you can create a complete ensemble of overlaid structures and just download it and then continue modeling with whatever you want. We also have some newer stuff I haven't shown you in the talk. So there's, for example, an activity finder. This is in the beta release. So we basically still working on it. So it has a complete link to the Campbell database. So these are hyperlinked. So you can basically go to the target, go to the ligand, and see measured activity values for these small molecules in the PDB file. Yeah, but this has to be taken with care. So if you start using it, so here are still some targets which, in our opinion, might not be the right one. So we have to find out what's going on here. But just to give you an idea that we do these things as well. So what we presented recently at the GCC, for those who have been there, was the Metalizer. And the Metalizer is a tool which allows you to analyze metal ions in here. And for example, we can look at a certain zinc. So what about this zinc here? So this one, so here is a zinc in the structure. And so what we can do now is we can estimate what kind of geometry we can, of coordination geometry we can expect for this zinc molecule. And here for this special zinc, I knew this already, I looked at them yesterday evening, something very strange happened. So in the back here, you basically see the typical geometries you see for this zinc ions, and you see the distribution in the PDB, and you see most of them are tetrahedral, but this one here as a preferred trigonal planar one. And if you look at it, you basically see it, you see that this is really odd, it's not really triangular. And now you probably already see the problem in the structure. Because if you look at the tetrahedral geometry, there's actually a missing binding partner, which would be okay if there would be space, but there is no space because there's this histidine in place, and this histidine might be oriented in the wrong way. Yeah, so if you would reorient the histidine, you would have a perfect arrangement of the zinc ions. So that would not only help you to find out what might be the most reasonable geometry here, it also helps you in one of the other cases to find an error. So I find this just yesterday, probably just by luck, by looking at a few structures. Okay, so that gives you an idea of what you actually can do. Of course, these things are linked and interactive. So you can, for example, look at the typical hydrogen bond distance or interaction distance, compare it to the distribution in the PDB. So the red one is the one we see in the structure, and actually work with these. Finally, just one thing I want to mention is a structure checker. This is also not completely done, but we basically have all the typical filter criteria which are used by the STIX dataset, by the iridium dataset, or by the platinum dataset, and combine this all to one tool so that you get a complete analysis of the protein ligand complex related to these properties. And here you see all these red flag boxes, which basically show reasons why this structure might not be part of, in my example now, the platinum dataset. Yeah, so for example, here you have very unusual bond lengths in the ligand structures, which might cause problems here. And at least this is a good reason to look at these. Okay, so this was just in a few minutes what the server can do for you. Usage is entirely free, and as you can imagine, every of these items would be a lot of work, and we continuously look for people who would like to do projects with us. So please feel free, if you have a bag of money, to get back to me. Okay, so we are already at the end, so I think the time is all mostly over, and I just want to add a few final remarks here to give you an idea why we do what we basically do. I don't want to repeat just what we have done. I think this is very well known. So what we always try to do is, so we try to combine precise chemical models with custom-made algorithms and machine learning. So that's actually the goal, yeah, because this is the counterpart of the simulation-based computational chemistry. We see that we do a data-driven combinatorial chemistry, and this is basically what we want to achieve. And in order to do this, we have to be very careful with data. That's what I learned in the past decade. So we need very high quality data collections for actually doing this kind of knowledge discovery from the data. So that's why we develop tools like EDR, why we take so much care of creating correct alignments, why we spend more than 3 years to actually get the protonations and protein ligand complex right, and so on. And the next thing is that we actually need methods to really get access to this data. We need good visual analytics platforms to browse information, because the amount of information becomes more and more. And this access by geometry I've shown you with Pelican is just a good example that we need good tools to actually get out the information by the questions we really have. I didn't talk so much about the ligand-based side, so I didn't talk about fragment spaces, creating molecules, etc. This is also a very big topic for us, but as time is always tight, so I didn't talk about this. But here we have very similar goals. We want to really represent what can be made, and rather that, and find out what is there, and really get new chemical space. We are always looking for efficient searching in all kind of problems we have, and we are also interested to get somehow the bridge between the intuition and the knowledge of medicinal chemists, and we want to put this into focus and use the computational tools to really support this for new ideas. So that's very much in the heart of our interest, and explains why we do what we do. Okay, so the picture is already old, but it shows several of the people who have done most of the work. IDEA is developed by Agnes standing over here. Stefan started with Protoss, and he also developed the alignment tool you've seen today. And yeah, so here's Therese. Therese developed the Pelican technology. And so, yeah, everybody of them somehow contributed, because we have a joint software development platform where everybody works together. Okay, I want to thank also Biosolve IT. We have a lot of exchange, not only ideas, a lot of exchange related to problems, but also exchange related to software. I want to thank all the funding partners, especially for Proteins Plus, which is very helpful for us. All right, with this, I'm at the end, and here are just a few links for you if you find it interesting. So that's the Proteins Plus link here in the center. Here above is a link which gives you access to the software, which can be downloaded from the ZBH web pages directly. It's free to use for academics. It's not entirely free for commercial customers, but you will see the regulations. It's not a big deal. And if you want to have something beyond academic software with good support, with great interfaces, you know where to find it at Biosolve IT. Okay, so that was everything from my side. I'm, of course, happy to answer questions which might be there in the list. I haven't looked at it while I was talking. So thank you for listening.
And thanking Matthias again here very much. I would like to point to the next webinar that we will be hosting in the new year. I will have the pleasure myself to introduce a new piece of software that we've written up during the last 2.5 years now, in collaboration with a specialist company on peptides, namely Zealand Pharma in Denmark. We have coined it PepC. Please save the date. It will take place on January 25th at 4 p.m. Central European Time. And as always, you'll be updated on biosolveit.de/webinar. And if you would like to get in touch with us here, then please feel free to write to contact@biosolveit.com. So, dear world, it was very exciting. We were widespread in terms of geographical locations. You were a great audience, very active, gave a lot of feedback. That's much appreciated. Expect a lot from us in the near future, also from us here at Biosolve IT. A bunch of these beautifully written and conceived tools from the Rarai group are available in CSAR that you can always download for testing from biosolveit.com/download. And with this, by the way, there is a Christmas animation now going on in CSAR. If you'd like to see that, go and download right away. And with this 5:30 now, I'll let you all free, and thank again. Hope to see you in the new year. Those of you who party Christmas, enjoy the festivities and get very safely into the next year. See you very soon. Thank you very much.